Hi guys, welcome back to the PhD Prep Lab channel. So we are back with another video on the Part B revision series for ICMR JRF. So today we are going to be covering basic physiology from animal physiology, and this is the part of the rapid revision for Part B of ICMR JRF examination, and this is going to be a one-shot video. So the core of this unit is blood and body fluids, digestive system, renal physiology, cardiovascular system, endocrinology, reproductive system. So these are the very important topics that I've highlighted in this slide. So this forms the core of this unit, and most of the questions on your basic physiology comes from these important highlighted topics mostly, along with other topics as well, which we are definitely going to be covering in this video. All the topics will be covered. The more importance are just highlighted here for your convenience. So before we move into the video, I want to remind you that we have uploaded more videos on this one-shot part B ICMR JRF revision series. So there is a video out on genetics and genomics. There is a video out on evolution, and there is another video on biosafety and bioethics. So all these are one-shot videos, and we do have an MCQ practice on biosafety bioethics video on biosafety bioethics unit, as well as those MCQs or practice questions are a bit difficult to find for biosafety and bioethics. So we have a separate video on that as well. So make sure that you check out all the videos. You can find the link in our description box for all the one-shot videos, and so that it will be easy for you to find the links and start your revision for your ICMR JRF examination that's coming up in that's coming up very soon. So without wasting much time, let's dive into the video. Let's start with the basics of physiology. So first of all, we have here is blood. So blood is a connective tissue. We all know about it, and it's composed of RBCs, WBCs, platelets, and plasma. Your plasma is the 93% composed of water with proteins. It maintains osmolarity and pH of the blood. And then we have serum, which is basically plasma without the clotting factor, specifically clotting factor 8. Clotting factor 8 is something that you have to remember. Now there is plasma protein synthesis, which is mostly in the liver. So it's the mostly the liver produced gamma globulins from lymphoid tissues. And you have on the left side the subunits of extracellular fluids along with their percentages. So you can read it out as well. So we'll move on to the next topic of hematopoiesis. So this is the continuous process of the making of all the types of blood cells from your hematopoietic stem cells within the bone marrow. So there is this multipotent hematopoietic stem cell, which is also called the hemocytoblast. So it makes two progenitor cells. One is the common myeloid progenitor cell and one is the common lymphoid progenitor cell, which gives rise to various number of different types of cells. So the myeloid progenitor cell will give rise to megakaryocyte, which develops into thrombocytes. Then it also forms erythrocyte, that's your red blood cells. Then your mast cells and your myeloblast, which has your WBC components right from your basophil, neutrophil, eosinophil to monocyte. Then on the lymphoid lineage, you have lymphoid progenitor cell, which forms the natural killer cell and the B and T lymphocytes. B lymphocytes mature and they develop into plasma cell. So this flowchart is very important because the lineages could be asked which cell is from myeloid lineage or which is from lymphoid lineage. So these are the kind of questions that are very frequently seen from physiology. And you have this chart where you can see the site of hematopoiesis. So during the fetal stage, during 0 to 1 month, it is mostly formed in the yolk sac, hematopoiesis, and then you see it in the liver as the fetus grows. And then after birth, it is mainly in the bone marrow, the site for hematopoiesis. Now, moving ahead, we have heme synthesis. So, this is a very important flow chart that you can see in this diagram. So, heme synthesis is mainly in the bone marrow. So, there is this step-by-step pathway. So, in the bone marrow, the heme is synthesized in the bone marrow, specifically in erythroblast and in liver also, which is in the hepatocytes. So the pathway of heme synthesis, it occurs in the mitochondria and partly in the cytosol as well. So first, it all begins with the formation of ALA, which occurs in the mitochondria, and the substrates to form ALA is the glycine and succinyl CoA, which you can see, and this forms your ALA. This is also the rate limiting step. So this is inhibited by heme by the feedback inhibition. Then from ALA, your PBG is formed, and then from PBG, so the formation of PBG is done in the cytosol, and from there the cycle down here from here continues in the cytosol. From PBG, your hydroxy methylbutylene is formed, which ultimately gives rise to coproporphyrinogen 3. Then the cycle goes back to the mitochondria, where you see the formation of protoporphyrinogen, then the formation of protoporphyrin 9, then finally the formation of heme, which is done in the mitochondria, and it is also inhibited by lead. So the summary, if I want to summarize it, from glycine and succinyl CoA, the substrates, it gives rise to ALA, then it forms PBG, then uroporphyrinogen 3, coporphyrinogen 3, protoporphyrinogen 9, then protoporphyrin 9, and then finally leads to heme. So it happens both in your mitochondria and partly in your cytosol as well. Now we have catabolism of hemoglobin. So it says how the hemoglobin is broken down. It is broken down mainly in the reticuloendothelial system (RES), especially in the spleen. If I want to put it out in simpler words, the site for catabolism of hemoglobin is spleen, and also in the liver and the bone marrow, where the old or the damaged red blood cells, they are destroyed. So first will be the breaking down of RBCs. So it happens when the RBCs are about 120 days old, and they are phagocytized by macrophages. The hemoglobin and is released, and it is split into globin and heme. So the globin, it is the protein part, and it is broken down into amino acids. These amino acids are reused for protein synthesis. Then the fate of heme. Then heme consists of iron and porphyrin ring. So the fate of iron and porphyrin ring, they are both different. First, iron is separated from the heme. It is stored as ferritin, and it is transported by transferrin to bone marrow for new hemoglobin synthesis. So iron takes part in new hemoglobin synthesis, and then there is porphyrin ring, which is converted into biliverdin, the green colored pigment, and this green colored pigment is reduced to bilirubin, which is the yellow pigment. Then bilirubin is unconjugated, it is indirect, and it is insoluble in water. Then what happens is this unconjugated bilirubin, it binds with albumin and it's transported to the liver. In the liver, in the hepatocytes, the bilirubin is conjugated with glucouronic acid by the enzyme UDP glucuronyl transferase. This forms the conjugated bilirubin, which is water soluble. Now, this conjugated bilirubin is excreted into bile and enters the intestine, where the intestinal bacteria converts it into urobilinogen, and this urobilinogen is converted into stercobilin, which gives the feces their brown color, and it is excreted out of our body with the help of the kidneys, and it also gives the urine its yellow color. So this is the this is the catabolism of hemoglobin, how all the protein part and the other parts, the heme, the globin, and in the heme, the iron and the porphyrin, how all of them they have different fates. So this is the flowchart on that, which is very, very important. A lot of questions have been seen from this. Now, moving ahead, we have hemostasis. So, there is this whole summary of hemostasis, which is basically hemostasis is the physiological process that stops bleeding at the site of injury while maintaining the normal blood flow. So, it happens in three major steps. First, after you have this any injury, any kind of injury, the blood vessels constrict. So, it undergoes vasoconstriction, where you see the reduced blood flow, mainly maintained by the endothelin, serotonin, and neural reflexes. Then you have the platelet plug formation, which is the primary hemostasis, wherein the platelets they adhere to the exposed collagen at the site of injury, and the platelets they become activated and release some kind of chemicals like your ADP and thromboxane A2. ADP and thromboxane A2 is important to remember. Then they aggregate the platelet formation. There is aggregation of platelet, which is the primary homeostatic and the formation of the platelet plug. Then there is the third concept, wherein a series of clotting factors are activated, the tissue factors. It activates the blood coagulation cascade, which finally gives rise to thrombin, which leads to fibrinogen and formation of fibrin. So, fibrin finally forms the stable hemostatic plug. So, there is this plug, plug formation after fibrin is formed. So in this, we have to remember that hemostasis, it mainly requires platelets, clotting factors, calcium, and vitamin K. So it prevents the excessive blood loss due to injuries. Now we have certain clotting factors which are important. You should remember these. So there is fibrinogen by the name factor 1. There is prothrombin, which is factor 2, and likewise. So this is the whole chart which you should try to remember because they sometimes directly ask the question that which is called factor 1, which is the factor 9, or which is the Hegman factor. Some questions like that have been seen. So you must remember this table. Now there is clot lysis process. So basically clot lysis is the process by which a formed blood clot is broken down after tissue has been repaired and restored to normal blood flow. So this process is also called fibrinolysis. So there is first the formation of plasminogen. So it is the inactive plasma protein incorporated into the blood clot during clot formation. So this plasminogen is converted into plasmin by plasminogen activators, which includes TPA, as you can see on the screen, UPA, and others, and a factor 12A, and something like kallikrein, etc., that you can all see. So these are the factors. Then plasmin is a powerful proteolytic enzyme. So this all leads to the action of plasmin. These all activators of plasminogen, they lead to the activation action of the plasmin, and the plasmin will digest the fiber threads, fibrin threads, fibrinogen and clotting factors, and this breaks the clot into fibrin. So when the clot is broken down into fibrin, it basically the fibrin it reduces to FTP. So during the removal of the clot, FDPs are cleared from the circulation. So once this FDP is cleared, the blood vessel lumen is reopened, restoring the normal blood flow. So to prevent excessive bleeding, this clot lysis process is very important, and this is the cycle how this inactivators and activators they all help in the clot lysis process. So now we begin with the cardiovascular system. So we know that the cardiovascular system includes the study of the heart and its diseases, and it is also known as cardiology. So the basics of heart, which um, how long it is, how small it is, it's mentioned on the screen, and the heart lies in the mediastinum, which is the anatomical region that extends from the sternum to the vertebral column. And these are the basics of the about the cardiovascular system, which you should have a look at. Now there are layers of the heart wall. So heart is consisting of three layers. Arranged from outer to inner is the epicardium, myocardium, and the endocardium. Endocardium is the innermost layer. So the superficial fibrous pericardium is composed of tough, inelastic, dense, irregular connective tissues. The middle layer is the myocardium, which is responsible for the pumping action of the heart. And the innermost layer is the endocardium, which is a thin layer of endothelial overlying a thin layer of connective tissues. Now, about the cardiovascular system's conduction system. So, the conduction system normally begins at the SA node. From the SA node, it travels, the action potential travels and it reaches to the AV node. From the AV node, the action potential enters the AV bundle, which is also known as the bundle of His. After propagating through the AV bundle, the action potential enters both the right and the left bundle branches. And from there, it reaches the Purkinje fiber, which rapidly conduct the action potential beginning at the apex of the heart upward to the remainder of the ventricular myocardium. Then the ventricles they contract, pushing the blood upward towards the valve. The SA node they sets the rhythm for contraction of the heart. It's also called the natural pacemaker. Now we have the cardiac cycle, which is basically the whole cycle of the cardiac, which is basically 60 to 80 beats per minute. How a healthy heart should act and how much it should beat. So during each heartbeat, which is a cardiac cycle, the heart contracts, which is also called as systole, and then it relaxes, which is also called the diastole. So there are certain stages of a cardiac cycle, which is the arterial systole, the contraction of the atria, the ventricular systole, which is the contraction of the ventricle, and there is the complete cardiac diastole, which is the relaxation of atria and the ventricles. So this is whole, the cardiac cycle lasts for zero point 0.8 seconds, along with the arterial systole for zero point 0.1 second, the ventricular systole for zero point 0.3 seconds, and the complete cardiac diastole for zero point 0.4 seconds. There is autonomic regulation of the heart, which means that the heart activity is regulated by the cardiovascular center in the medulla oblongata, which is under the neural regulation of the heart. Then the autonomic control is the cardiovascular center just adjust heart rate by altering sympathetic and parasympathetic nerve impulse of the ANS. Then there is hormonal regulation also, which is controlled by the norepinephrine and epinephrine, which is from the adrenal medulla, which increases the heart rate and pumping efficiency. There is ionic regulation of the heart rate as well, which is the intracellular and extracellular cations, especially sodium and potassium ions, which influence the cardiac excitability and the rhythm. Now, there is cardiac output, which is the volume of the blood pumped by each ventricle into the aorta or the pulmonary trunk per minute. There are baroreceptors, which detect the increase in the atrial blood pressure, and there are chemoreceptors, which senses changes in the blood, like changes in CO2 levels, O2 levels, pH, indicating the tissue perfusion status. Now there is electrocardiogram, which is also called ECG, which is a graphical recording of the electrical activity of the heart obtained from the electrodes placed on the body surface. This you must have seen, its reports you must have come across. So these looks like some waves. So the P wave here represents the atrial depolarization, the QRX complex represents the ventricular depolarization, and the T wave represents the ventricular repolarization. So which wave represents what is somewhat very important. And the stroke volume is the volume of the blood which is ejected by ventricles during each contraction. Now we come to gastrointestinal physiology, and before we move to the digestive system, I want to remind you that all the notes you can find, and you have to join our Telegram group for that, where you can find all of these notes and the notes of other one-shot videos as well. So make sure that you stick to the end of the video and get the QR scanner to scan and join our Telegram groups for notes and for internship and workshop updates. So we have two different channels. So now continuing to the digestive system, first we have the gastrointestinal physiology, where you eat the food, goes into the oral cavity, the salivary glands, they activate the food, it then pushes down through the pharynx and the esophagus, goes to the liver, and then the small intestine, gallbladder, pancreas, they all secrete and act on the mixed food, then it goes to the large intestine and exits from the rectum. Then you have the overview here where we see that the salivary glands, they secrete amylase, mucus, lysozyme. The esophagus, it secretes mucus. Stomach will secrete gastric juices which consist of HCl, pepsin and mucus. Then you have small intestine where you see under pancreatic enzymes there is amylase, maltase, trypsin, chymotrypsin, lipase, nuclease, and then you have large intestine which secretes mucus, vitamins, and gases, and it is enteric bacteria. Where you can find enteric bacteria is your large intestine. So the secretions are important. Which secretions consist of which and which organs respectively secretes it is important. Now we move to endocrinology. So first up, we have the hypothalamus, which is the master of the master glands. So it has certain secretions. So basically, it stimulates the anterior pituitary hormone secretion. So it has secretions like TRH, which is the thyrotropin releasing hormone. It stimulates TSH. Then we have CRH, which is corticotropin releasing hormone, which stimulates the ACTH, which is also the secretion of the pituitary. Then you have GnRH, which is gonadotropin releasing hormones, which stimulates FSH and LH. So this is the chart where you can see that which of the hypothalamic hormone secretes which of the pituitary hormone. So all those releasing hormones are mainly secreted by hypothalamus, as you can see on the screen. Then we have certain other glands as well. We have adrenal glands, which regulates the metabolism of blood pressure, immune system, and stress. The hormone that adrenal glands secrete are adrenaline, cortisol, aldosterone, DHEA, testosterone. So adrenal glands are found above the kidneys. Then we have pancreas. So these secrete insulin, glucagon, somatostatin. Then we have ovaries and testes, which secrete the sex hormones. Then we have thyroid gland, which secrete T3, T4, and calcitonin. The pineal gland will secrete melatonin, which regulates the sleep-wake cycle. Then the pituitary gland, which secretes FSH, FLH, prolactin, growth hormone, TSH, ACTH, and then finally hypothalamus, which stimulates the secretion of the hormones of the pituitary gland, as I mentioned before, and you can see on the chart as well. So here is the adrenal gland and its details, the anatomy and the hormones it secretes. So we have discussed which hormones it secretes. Now we have the anatomy of it. So you can see that the adrenal glands, they are paired and they are triangular endocrine glands. They are located on top of each kidneys. So they have certain anatomical uh, zones, which are first of all the zona glomerulosa, which secretes the hormone aldosterone, which is a mineralocorticoid, which regulates the sodium potassium level and the blood pressure. Then we have zona fasciculata, which secretes the cortisols, the glucocorticoids. Then we have the zona reticularis, which secretes androgens, that is DHEA. So, which zone of the adrenal cortex secretes what is important. So, all of these zones are under adrenal cortex. So, adrenal cortex is the outermost layer of the adrenal glands. Then we have adrenal medulla, which is the inner layer. So, it is composed of chromaffin cells, which is the modified postganglionic sympathetic neuron. It produces catecholamines, which secretes the adrenaline, that is epinephrine, and the noradrenaline, which is the norepinephrine. So epinephrine, which will increase the heart rate and the blood pressure and the blood glucose, and the norepinephrine is responsible for vasoconstriction. So these points are important. And certain other functions of the adrenal gland is also that the cortisol that it secretes is a stress hormone. Then the aldosterone, it regulates the sodium and water retention. And there is adrenal androgens, which is there in the puberty. And there is catecholamines, which is basically your adrenaline and non-adrenaline, which are also the fight or flight response hormones. Then we have your thyroid gland. The thyroid gland is shaped in the form of a butterfly. It is also an endocrine gland and it is located in the front of the trachea and below the larynx. So it regulates the metabolism, growth, and the development of a human body. So the hormones that the thyroid gland it secretes is T3, which is the triiodothyronine, and the T4, which is the thyroxine. These are produced by the follicular cells from tyrosine and iodine. It regulates the and metabolic rate and promotes growth and development. Then there is calcitonin, which is produced by the C cell, which lowers the blood calcium levels and inhibits osteoclast activity. Now we have pituitary gland. So this pituitary gland is a small pea-sized endocrine gland which is located in the stella turcica of the sphenoid cord, and it is connected to the hypothalamus via the pituitary stalk. And it is also called the master gland because it regulates several other endocrine glands as well. So there is anterior pituitary and posterior pituitary. Anterior pituitary is the area of hypophysis. It's composed of glandular epithelial tissues and it secretes growth hormone, TSH, ACTH, FSH, LH, prolactin, MSH, which is the melanocyte stimulating hormone. And the functions are discussed previously. And there is posterior pituitary as well, which is also called the neurohypophysis, which secretes ADH, which is vasopressin, which helps in water retention, and oxytocin, the other hormone, which is responsible for uterine contraction and milk ejection. So these are ADH and oxytocin is secreted by posterior pituitary. Then we have the respiratory physiology. So here you can see a lot of information has been given on respiratory physiology. So it all starts with the nasal cavity. So first, your respiratory physiology conducts of two zones. First is the conducting zone, which conducts the air, and then is the respiratory zone, which is responsible for gas exchange. So for your conducting zone, you have nasal cavity, pharynx, larynx, trachea, and bronchi. And then in the respiratory zone, we have respiratory bronchioles and alveoli. Now, alveoli are also of two types, the type 1 and there is type 2. So type 1 cells are the flat epithelial cells responsible for gas exchange. The type 2 cells, they produce a type of surfactant which is helps the membrane not to collapse, the respiratory membrane not to collapse. And the respiratory membrane is made of alveoli and the capillaries. Then you can see about the pulmonary circulation and innervation. So the pulmonary circulation is basically where the deoxygenated blood from the is carried from the heart to the lungs and returns on the heart and the lungs. There is exchange of O2 and it returns the oxygenated blood back to the heart. So this is your pulmonary circulation. Now we have a very important concept of oxygen hemoglobin dissociation curve. So this curve is very important. It is a sigmoid type of graph which shows the relationship between partial pressure of oxygen and hemoglobin saturation. So this is sigmoid shape due to the cooperative binding of one of the oxygen molecules, which increases affinity of the hemoglobin for the next oxygen. In the plateau phase, the hemoglobin is nearly saturated. It ensures oxygen delivery even if the partial pressure of oxygen slightly dropped. Then at the other phases, you can see which shape of the graph is responsible of due to is acting due to which action. So you can see that in the lower arrow, you can see that as the oxygenated blood reaches the tissues, a small decrease in the partial pressure of oxygen causes a large decrease in the saturation, which is when the oxygen is released. So there are certain factors that affects this oxygen hemoglobin dissociation curve. So there is a right shift, which means the decrease in the affinity, which means that there is easier oxygen release. So the decrease in the affinity, the right shift will be caused due to increase of carbon dioxide levels, increase of H plus ions, which means the lowering of the pH, and increase of temperature and increase of 2,3 bpg. Then you see the left shift, which happens due to the increase in affinity, which means that um, it is not possible for the oxygen to release now. The affinity gets stronger. So the affinity gets stronger means the left shift happens due to decrease in carbon dioxide levels, increase in pH, or decrease in temperature, and decrease in 2,3 ppg. So the right shift basically enhances oxygen delivery to active tissues, and the left shift occurs in fetal hemoglobin, which maximizes oxygen uptake from the maternal blood. Now we come to renal physiology. So you can see that the most important or the basic unit, the functional unit of the renal system, which is of the kidney, is the nephron. So the cells of the kidneys are called nephrons. Now, there are certain parts of nephrons which includes the glomerulus, and there is then Bowman's capsule, which is surrounding the glomerulus, and there is PCT, the proximal convoluted tubule, and then loop of Henle. Then there is DCT, which is the distal convoluted tubule, and there is efferent arteriole, which is the point of entry, which of the unfiltered blood, and there is efferent arteriole, which is the point of exit of the filtered blood. Then there are peritubular capillaries, which surround the proximal and distal convoluted tubules for immediate resorption of necessary substances that are needed for the circulation. Then you have vasa recta, located adjacent to the ascending and descending loop of Henle. Now you have two types of nephrons. First is cortical nephron, then there is juxtamedullary nephron. The cortical nephrons are in majority, which is 85%, which are responsible for removal of waste products and reabsorption. The juxtamedullary nephron is primarily for the concentration of the urine. It concentrates the urine. Then the renal function, you can see that's homeostasis, excretion of waste, maintenance of blood pressure, and erythropoiesis. Then you have renal blood flow, which is happening in the renal artery, which supplies blood to the kidney. The kidneys receives a large amount of blood, and based on the average body size, the total blood flow should be 1200 ml per minute, and total renal plasma flow should be 600 to 700. Now, the most important topic of the renal physiology is the countercurrent mechanism. So, it is the process where the kidneys help to concentrate the urine. So, the more concentrated the urine is, the more filtered the blood is. So, basically, in the countercurrent mechanism, the concentration of the urine is done by creating a gradient for water reabsorption in the loop of Henle. So it happens all in the loop of Henle. So the principle behind this is the flows of the filtrate and the blood move in the opposite direction in the countercurrent mechanism in the nephron and vasa recta. This maximizes the solute and water exchange and establishes a hyperosmotic medullary interstitium. So the descending limb is permeable to water and impermeable to salts. So the water level leaves here by osmosis. Then the ascending limb is impermeable to water and actively pumps out sodium, potassium, and chlorine ions, which makes the medulla hyperosmotic. The result is the osmotic gradient from the cortex to medulla has been formed, which enables the water reabsorption from collecting ducts. So the blood in the vasa recta flows opposite to the filtrate in loop of Henle, and it prevents the dissipation of medullary gradient while allowing nutrient and waste exchange. So all of this leads to production of a concentrated urine, and it conserves water. It maintains the electrolyte balance as well. So this is how the regulation of aldosterone secretion by the renin-angiotensin-aldosterone is done, which is the RAA pathway. So renin-angiotensin mechanism is the hormonal system which regulates the blood pressure, fluid, and electrolyte balance. So first, what happens is there is the release of renin, which is produced by the juxtaglomerular cells of the kidney in response to a lower blood pressure or lower sodium concentration in the distal tubule, and there is angiotensinogen conversion. Renin converts the angiotensinogen from the liver to angiotensin 1. This angiotensin 1 is converted to angiotensin 2 by the action of ACE, which happens mainly in the lungs, which converts the angiotensin 1 to angiotensin 2. This angiotensin 2 leads to vasoconstriction, which leads to an increased blood pressure. The blood pressure here then goes back to normal. It stimulates the aldosterone secretion from adrenal cortex. Sodium and water retention is there, which increases the blood volume. So this stimulates the ADH release from the posterior pituitary, which leads to water resorption, which stimulates thirst and water intake, hence is increased. So all of this, to summarize, is basically due to a lower blood pressure. The juxtaglomerular cells they act and release renin. Renin converts angiotensinogen to angiotensin 1, and then on ACE will act on angiotensin 1 and convert into angiotensin 2, which leads to vasoconstriction plus aldosterone plus ADH secretion plus thirst, which altogether increases the blood pressure and blood volume. Now there is reproductive physiology. We have the male and the female reproductive system, and the XY means male and XX means female. The XY has SRY genes, and the formation of testis is mainly due to anti-Mullerian factor, and in ovaries there is no anti-Mullerian hormone or no testosterone. The female reproductive physiology is seen here. So in female reproductive physiology, there is complex hormonal involvement, and we have an ovarian cycle which lasts up to 28 days, which is divided into follicular phase, ovulation, and luteal phase. So first in the follicular phase, the FSH it acts on growing the ovarian follicles. The follicles secrete estrogen, which thickens the endometrium. Then the ovulation occurs at around 14th day due to LH surge. So it's important to remember when the LH and FSH levels will be increased and at its peak, and when they will be decreased. So during ovulation, the LH level is at its peak. Then mature Brachyn follicle, it ruptures and releases the ovum during ovulation. It is triggered by LH surge. Then in luteal phase, the ruptured follicle, it secretes progesterone and prepares the endothelium for implantation. Now there is the uterine cycle also, in which it starts with the menstrual phase. There is the where the shedding of the endometrium occurs in form of menstruation. Then there is proliferative phase, which is stimulated by estrogen, in where the endometrium thickens. The estrogen is responsible to thicken your endometrium layer. And then there is the secretory phase, which is stimulated by progesterone, in which the endometrium becomes secretory for implantation of the zygote. Now, at last, we have male reproductive physiology, in which you can see a lot of hormones are involved again. So, you can see on this chart the key hormones, which is responsible and the source of it and the effect of it. So, you can see LH also is working in the male reproductive system, which is important, and is secreted by the pituitary gland from the anterior lobe. And the target is intestinal cells, and it releases testosterone. So, LH releases testosterone, and testosterone is responsible for your spermatogenesis and vascularization, and it has a negative feedback mechanism which inhibits the release of GnRH and LH. Then there is ABP, androgen binding protein, which keeps the testosterone levels elevated. Then we have inhibin also, which inhibits FSH release. And there are now the primary and secondary sexual characteristics. The example are the enlargement of penis, scrotum, testicles, and hair growth, etc. So you can see on the screen all the important details are mentioned. So make sure that you revise all of the slides well. And this was it from this basic physiology part. So we hope that you like the video. If you have suggestions, any of it, so you can leave it in the comment box. And do follow our Telegram channels. There are two separate groups. So first is the Life Science JRF notes, where you can find notes for all the important topics, all the videos that we have uploaded, all the notes from there. Also, you can find it on this Telegram channel. So make sure that you join this. And for internship, job opportunities, and workshops, we have another Telegram group which you can join to get alerts and instant updates on internships and workshop opportunities. And make sure that you have watched our previous one-shot videos as well, which is on part B ICMR JRF revision. So you'll find all the links of all the videos in the description box. So do check it out. And if you have any doubts or suggestions, leave that in the comment box and share it with your friends so that they also can prepare better for their ICMR JRF examination. And we'll see you with another video very soon for part B JRF revision. Until then, like our video, share with your friends. Make sure that you have subscribed to our channel and press the bell icon so that you don't miss on future coming videos. And thank you for watching.